A plant that should have stayed a weed, energy is being locked underground in a form that moves too slowly to justify its own existence. Calories sealed inside living tissue that resists urgency, resists speed, resists extraction, and yet it persists. The outcome should be decay, stagnation, evolutionary irrelevance. Instead, something holds. A contradiction stabilizes. Long before fields, before records, before any concept of engineering, a wild morning glory experienced a genetic intrusion that biology normally treats as a fatal mistake. Foreign DNA crossed a cellular boundary it was never meant to cross, not through breeding, not through selection, but through infection. The vector was a soil bacterium, agrobacterium, an organism that survives by forcing plants to grow tumors, hijacking their cells into producing compounds only the bacterium can use. In most cases, this relationship ends with damage, metabolic chaos, and a weakened host. But in this lineage, the transferred genes did not vanish. They integrated, they stabilized, they began to be expressed as if they belonged. Over generations, that infected plant did not collapse. It reorganized. Storage tissue thickened below ground, starch accumulated where none was required before, energy was hoarded in silence. This was not domestication. No human guided it. No selection pressure asked for yield or taste. It was a molecular accident that survived long enough to become permanent. Today, those bacterial genes are still there, embedded in every sweet potato genome ever sequenced, functioning, inherited, unquestioned. By modern definitions, this is a genetically modified organism created without consent, without oversight, without reversal. The unsettling part is not that it happened, but that it worked. The plant became something heavier, slower, more conservative with energy than its relatives, converting sunlight into dense carbohydrate reserves that remain inert for months. From a survival perspective, this is inefficient. Roots that large demand resources, invite predators, slow reproduction. From a systems perspective, it is worse. Energy that cannot be rapidly mobilized becomes dead weight under pressure, and yet this structure persisted across climates, across continents, across human history. People later found it and learned to eat it, mistaking usefulness for intention. But the transformation was never for us. It was the residue of an ancient infection that didn't finish the job. What exists now is not optimized, not elegant, not scalable. It is a compromise that never resolved. A weed that should have remained thin and disposable instead became a subterranean battery with no off switch, proof that biological rules do not enforce comfort or efficiency, only survival past the next failure point. Something entered this plant that should have destroyed it. The plant absorbed the error, rewrote its boundaries, and continued, carrying the mark forward for 8,000 years. Not because it was better, but because nothing stopped it. Multicellular life is designed to defend its borders, cell walls, membranes, nuclear envelopes, all meant to slow change to a pace evolution can survive. Genes are supposed to move vertically, parent to offspring, filtered by reproduction, diluted by time. Anything faster threatens collapse. Horizontal gene transfer breaks that rule. In bacteria, it is routine, almost casual. In complex plants, it is a structural risk. When foreign DNA inserts itself directly into a stable genome, the expected outcome is misregulation, developmental failure, sterility. The system should reject it or die trying. But agrobacterium does not ask. It injects genetic material into plant cells as a survival strategy, forcing the host to build nutrient-producing tumors. This is not cooperation. It is biochemical coercion. In most plants, the damage remains localized and temporary. In the lineage that became the sweet potato, something different happened. The transferred genes crossed into germline tissue, the one place they were never supposed to persist. Once there, they avoided silencing. They avoided deletion. They began to replicate as the plant reproduced, copied alongside native genes as if they had always belonged. This is the biological equivalent of a manufacturing defect propagating through every future model. The paradox is not that the genes remained, but that they became functional without dismantling the rest of the system. These bacterial sequences encode proteins involved in hormone signaling and cell growth, the same pathways that control root development. Instead of uncontrolled tumors, the plant produced in large storage roots, organized, vascularized, metabolically integrated. The margin between pathological growth and useful structure is thin. Cross it unintentionally, and the organism becomes unviable. The sweet potato crossed it and stabilized on the other side. This should not be repeatable. In laboratory settings, agrobacterium-mediated transformation requires controlled conditions, selectable markers, careful screening. In nature, there is no quality control. Most attempts fail silently. This one did not. The result is a plant carrying bacterial DNA that modern agriculture would classify as transgenic. Yet it emerged without selection for yield, without human preference, without a safety margin. The genes remain active today, detectable, measurable, still expressing proteins of non-plant origin. They do not improve efficiency. They do not maximize output. They simply do not break anything badly enough to be removed. Evolution did not optimize them. It tolerated them. That distinction matters. Tolerance is not endorsement. It is survival under constraint. The sweet potato exists because its genome absorbed an intrusion and found a configuration that did not immediately fail. There was no foresight, no advantage guaranteed, only a narrow path where damage did not exceed repair capacity. This is the uncomfortable truth of horizontal gene transfer in complex life. Catastrophic change is not required. Slightly wrong can be enough to persist forever. A foreign instruction set entered a plant, altered its architecture, and remained, not because it was ideal, but because the system could endure it without tearing itself apart. Large reserves create the illusion of abundance, but biology does not measure food by volume. It measures timing. Energy that cannot be accessed at the required rate is functionally absent. The sweet potato is dense with carbohydrates, primarily starch, packed into swollen roots that can account for the majority of the plant's biomass. On paper, this looks efficient. In practice, it is restrained. The starch granules are structured to resist rapid breakdown, and the sugars released during digestion enter the bloodstream more gradually than those from cereals or processed tubers. For the plant, this slow mobilization reduces metabolic shock and stabilizes growth under fluctuating conditions. For humans, it creates a mismatch. Societies that rely on staple crops are constrained by how quickly calories can be converted into labor, heat, and reproduction. Potatoes, rice, and wheat deliver energy with fewer delays. Sweet potatoes do not fail nutritionally, but they hesitate. This hesitation becomes critical at scale. A population under stress does not need calories eventually. It needs them now. The paradox is that the same property that makes sweet potatoes metabolically stable makes them structurally unsuitable as a dominant industrial staple. The storage routes evolved as buffers, not engines. They smooth out scarcity, but they do not accelerate output. Even their glycemic response, often framed as a health benefit, reflects this limitation. Energy release is capped by design. There is no mechanism to overclock the system without breaking it. For the plant, rapid starch turnover would destabilize growth and invite collapse under drought or nutrient stress. For human systems, that caution translates into reduced immediacy. In famine conditions, speed matters more than total yield. This is why sweet potatoes historically appear as supplemental staples, emergency crops, or regional mainstays rather than global anchors. They sustain life, but they do not amplify it. The discomfort comes from recognizing that abundance without velocity is not security. A field full of sweet potatoes represents stored potential that refuses to rush. Nature did not design this organ to serve continuous extraction. It was designed to wait. In biological terms, waiting is often safer than acting. In civilizational terms, waiting is expensive. The sweet potato occupies that narrow zone where energy exists but resists urgency, where calories are present but arrive too late to fuel expansion. It is not a failure of nutrition. It is a failure of timing. A system that hoards energy and releases it cautiously survives longer, but it does not scale cleanly. The plant did not evolve to feed cities. It evolved to outlast conditions that would kill faster, leaner competitors. That success comes with a cost that only becomes visible when the system is pushed beyond subsistence into acceleration. Survival across environments is often mistaken for strength, but in engineered systems, flexibility behaves like noise. The sweet potato tolerates heat, irregular rainfall, poor soils, and biological stressors that would reduce more specialized crops to failure. Its physiology absorbs variation rather than amplifying it. Roots form at inconsistent depths, vines adjust growth direction opportunistically, storage tissue responds to microconditions rather than uniform schedules. In nature, this is protective. In industrial agriculture, it is disruptive. Large-scale food systems depend on predictability, synchronized maturation, standardized size, and mechanical compatibility. The sweet potato resists all four. Individual plants of the same cultivar can produce roots that vary widely in shape, density, and timing of development, not because of error, but because the plant's regulatory networks are tuned to local feedback rather than centralized control. This decentralization keeps the organism alive under uncertainty, but it undermines efficiency when imposed on monoculture. Harvest machinery designed for uniform tubers encounters irregular geometry. Storage systems optimized for consistent respiration rates face uneven spoilage risk. Processing lines calibrated for starch content encounter variation that exceeds acceptable tolerances. None of these failures are dramatic. They accumulate quietly. Each deviation increases energy cost, labor input, waste. The paradox is that the plant's evolutionary success creates systemic friction when scaled. Its genome favors resilience over obedience. Potatoes, by contrast, collapse quickly outside narrow conditions, but within them, they behave. They synchronize. They submit to optimization. The sweet potato does not. Attempts to breed uniformity reduce stress tolerance. Attempts to maximize yield reduce environmental range. Each push toward standardization erodes the very properties that made the plant reliable in marginal systems. This is not a technical limitation that can be solved with better tools. It is a biological trade-off encoded at multiple levels, from hormone gradients to root initiation patterns. The plant's flexibility distributes risk internally, but industrial systems externalize risk onto infrastructure. When those two logics meet, something gives. Usually, it is the crop's role in the system. This is why sweet potatoes excel in subsistence and smallholder contexts, where variation can be absorbed socially, but struggle to dominate supply chains built for speed and sameness. The discomfort lies in recognizing that adaptability, often framed as evolutionary virtue, becomes a liability under artificial selection regimes. The sweet potato is not inefficient. It is incompatible. It exists in a state that prioritizes survival across uncertainty rather than performance under control. That choice was made long before humans noticed it, and it cannot be undone without dismantling the organism itself. Feeding large populations is not only a question of yield, but of compliance. Staple crops that dominate global systems share a property beyond nutrition. They submit to logistics. They can be harvested on schedule, stored with predictable loss curves, transported without rapid degradation, and processed into interchangeable forms. Sweet potatoes violate this quietly. Their storage routes remain biologically active after harvest, respiring, converting starch to sugar, responding to temperature and humidity with metabolic adjustments that resist dormancy. This activity shortens storage windows and complicates transport. Unlike grains, they cannot be dried into inert units without transformation. Unlike potatoes, their post-harvest physiology varies enough to demand constant monitoring. Industrial systems tolerate narrow bands of behavior. Sweet potatoes operate outside them. The paradox is that at the household or village level, this is not a problem. Consumption matches harvest. Loss is absorbed locally. At scale, it becomes structural. Refrigeration slows metabolism but increases chilling injury risk. Warm storage reduces damage but accelerates respiration. Each solution shifts the problem rather than removing it. Processing into flour or starch requires additional energy inputs that erase the crop's apparent efficiency. The calories are real, but they refuse standardization. This resistance is not intentional. It emerges from the same metabolic conservatism that allowed the plant to survive an ancient genetic intrusion. The system prioritizes internal stability over external demand. Human food networks prioritize the opposite. When these priorities collide, one side adapts or exits. Historically, sweet potatoes remained regionally critical yet globally peripheral, sustaining millions without ever becoming a universal backbone. This is not because they are inferior, but because they do not conform to the rhythms of industrial consumption. They feed bodies, but they resist pipelines. The unsettling realization is that abundance alone does not guarantee dominance. A crop can be nutritionally sufficient and still fail as infrastructure. Sweet potatoes expose the hidden requirement of modern food systems: obedience at every stage, from growth to decay. Any organism that insists on responding to its own internal signals rather than external schedules becomes a liability. The sweet potato continues to exist at the edge of this tension, valuable, reliable, and yet structurally incompatible with the systems that claim to optimize survival at scale. Part 6. An accident that refuses to scale. The sweet potato exists because a boundary was crossed once, accidentally, and then never crossed again. The bacterial genes embedded in its genome did not create a platform for endless improvement. They created a narrow configuration that worked just well enough to persist. This is the final paradox. Modern genetic engineering assumes modularity, that traits can be added, adjusted, optimized incrementally. The sweet potato contradicts this assumption. Its defining feature is not a single beneficial gene, but a fragile equilibrium between foreign DNA, plant regulatory networks, and environmental tolerance. Alter one parameter too aggressively, increase starch density, accelerate growth, enforce uniformity, and the system destabilizes. Yield gains invite disease pressure. Uniformity reduces resilience. Faster growth strains vascular transport. Each improvement attempt approaches a failure threshold the organism cannot cross safely. This is why the ancient genetic accident cannot be cleanly repeated or extended. It was not engineered with margins. It has no redundancy designed for optimization. The plant survives because nothing has pushed it far enough to break. This places it in an uncomfortable category, a successful outcome that resists improvement. Agriculture prefers systems that respond predictably to intervention. The sweet potato responds unpredictably because its origin was not predictive. It is the fossil record of a mistake that happened to endure. From a systems perspective, it represents a dead end that remains alive. It feeds people, but it does not invite acceleration. It offers calories, but not scalability. Its genome carries proof that drastic change does not require intention, but also proof that not every successful change can be safely repeated. The unsettling truth is that some of the food humanity depends on exists not because it was optimized, but because it avoided catastrophe by chance. The sweet potato is not a triumph of design. It is a survivor of error. And systems built on survival by accident do not expand gracefully. They persist quietly, resisting correction, reminding us that biology does not promise progress, only continuation past the last collapse point.